Next, I'd like to introduce Dr. Chris Pierce. He's a professor of neuroscience in the Department of Psychiatry. He received his bachelor's degree in psychology from the University of Kentucky and his PhD in neuroscience from Indiana University, and where he did his work on dopamine signaling in the brain. Chris did his postdoctoral work at the University of Washington, where he focused on animal models of psychostimulant addiction. Currently, his lab studies the cellular and molecular mechanisms underlying cocaine relapse in animal models. And he has a highly impressive publication record, and recent work from his lab, some of you may have seen, has been featured on the Colbert Report. So let's give a warm welcome to Dr. Pierce.
So thanks, Leonardo. Thanks to all you for showing up. This is really terrific. So I really am going to talk about cocaine tonight, but the first bit of the talk is actually going to be history. It's not going to be science history, but it's going to be history, but trust me, it's not going to be what you're expecting, but it is going to be relevant. And my story goes way back to 1944, and in September of that year, World War II in Europe has reached its apex. Allied forces have liberated France, Belgium, and southern Holland. And a massive Allied assault is planned to liberate the north of Holland. This operation is termed Operation Market Garden, and at that time it was the largest airborne attack ever staged. It took place between September 17th and 25th in 1944. It was a spectacular failure. In response, Hitler punished the Dutch, who collaborated with the Allied invaders. Germany instituted a complete blockade, including all food and fuel shipments. The Dutch referred to the winter of 1944-45 as the Hunger Winter. Unfortunately, winter came early that year and was particularly bitter. Food stocks were rapidly depleted. In October of 1944, 4.5 million people in and around Amsterdam began to starve. The famine lasted until Holland was liberated at the end of World War II in May of 1945. The people in North Holland ate tulip bulbs and stripped their homes for firewood. Tens of thousands died from malnutrition and exposure. So at that time, when all the food ran out, I mean, they managed to sneak in rations from Sweden occasionally, though another problem was that the harbor froze, so they couldn't get things in with boats. But the little food that they had, the Dutch systematically rationed, and the ration was 450 calories of food per person per day. And there are drinks at Starbucks who have more than 450 calories in them. This is about a fifth of what the average American eats per day. This person is a 16-year-old young girl who survived the Hunger Winter. Her name was Audrey Hepburn. Audrey Hepburn suffered from various physical abnormalities, sicknesses throughout her lifetime, which she attributed to living through the Hunger Winter. And her experience in the Hunger Winter undoubtedly influenced what she did when she retired from being an actress. She became an advocate for famine victims throughout the world and was as an ambassador for UNICEF. So despite the suffering in the Hunger Winter, detailed medical records continued to be compiled. And this provided a unique opportunity to study the effects of famine on human health. And the babies born during and just after the Dutch famine have been studied for their entire lives. And this is two studies here. So one of the early ones in 1972 in Science Magazine, and another one here from more recently in 2006. But there have been dozens of studies examining these children who were in utero at the time of the Hunger Winter, as well as their children and their children's children. So there's a huge database now. So a few of the observations just quickly, because this isn't directly relevant to my talk today, but the children whose mothers were malnourished only early in pregnancy had higher obesity rates, as well as other health problems, including diabetes, which is undoubtedly related to the obesity, as well as some psychiatric problems such as schizophrenia and depression. So if your mother was malnourished and you were in her uterus at that time, when you grew up, you were more likely to have problems with obesity, schizophrenia, and depression. And this isn't too horribly surprising because your mother was exposed to the famine, but so were you. I mean, you were inside her body at that time. But then more unexpectedly is these health problems, these very same health problems also plagued the grandchildren of the mothers who were malnourished in early pregnancy. So this is unexpected and might tell us about some interesting underlying biological processes, which is what we're going to be talking about today. So we think about inheritance as being a genetic process. And a genetic process obviously involves a change in DNA, which usually takes place across millennia in the Darwinian scheme of things, as I show on this next bullet point. But the environmental changes, even as severe as living through a famine might be, that's not enough to produce changes in the DNA in a person's egg or sperm, thus to make these changes heritable. In fact, there's very few things that are capable of doing that. There are some things, you know, these poor people in Japan when the nuclear power plant melted down. I mean, that's one of the examples, and there are some nasty chemicals that we know of that can cause changes in the DNA sequence, but that's about it. So most of the other things in the environment don't cause changes in the DNA sequence. So any of these changes that we're talking about in the grandchildren must be through a different process, and that process is known as epigenetic inheritance. So for the purposes of this talk, this is both the process and the mechanism. We're really not going to talk about the mechanism today. But epigenetics, just what it literally means is above genetics or in addition to genetics. Okay? So this is all a prequel to what I'd like to talk about today. So what my lab studies, as Leonardo said, and Leonardo is a member of my laboratory, is we study animal models of cocaine addiction. So this literature indicating that we might have these transgenerational effects of environmental exposures got us to thinking about, well, what about cocaine? What about drugs of abuse? I mean, might they influence the offspring and then the grand offspring and generations beyond that? So we studied paternal cocaine exposure. So the dads took cocaine, and we looked at learning and memory in the offspring. So here we have the setup over here on the left. So this is the standard male Sprague Dawley rat in what we call an operant chamber. And up here we have a syringe that's full of cocaine. I don't know if you can see it, but there's a line that goes up like this, and it goes down to a port between the guy's shoulder blades. And from that port, the catheter goes over his shoulder into his jugular vein. So we have a direct line from this syringe of cocaine to the animal's heart. And we allow this animal to press this lever to receive cocaine. Now, how hard do you think it is to get them to press that lever for cocaine? It ain't hard at all. We put them in the box, we close the door, we don't do anything, no kind of pre-training. They explore around the box, they bump into the lever. Once they get a few shots of cocaine, they know it, they like it, they press a lot for it. So in this experiment, we have the cocaine dads and we have the saline dads. So this is the cocaine self-administration behavior, and these guys self-administered cocaine 2 hours a day for 60 days. How much cocaine is this? It's a lot of cocaine. So as a matter of fact, we have to regulate their cocaine intake because the advertisements you see on TV are true. If we left these guys to their own devices, every single one of them would overdose on the cocaine and die. Every single one of them. The saline control animals, every time this guy received an infusion of cocaine, there was another box down here, and they had a guy set up just like this, but hitting the lever did nothing, and just every so often, when this guy received cocaine, this guy down here received saline. So that was our control group. Okay, so we took these cocaine guys and we took the saline guys and we mated them to naive females, okay? So in science, so these are the parents. So we call them the parents, scientists call it F0, the F0 generation. So the cocaine guys and the saline guys were with the females just long enough to impregnate them, and then the cocaine guys went away and were never seen again, just like cocaine addicts in real life. So the females got pregnant, they had their babies, and the babies grew up, and then we asked the babies various questions. Well, first off, what do we call the babies? So this generation, the parents are called F0, and scientists call the children F1, the first generation. The children is F1, and if your dad took cocaine, we called you cocaine-sired. If your dad received saline passively, we called you saline-sired. Once these guys grew up and reached adulthood, we asked them, we didn't ask them, we looked at learning and memory problems in these guys. And we used two different tasks. one of which is called object place recognition, which is dependent on an area in your brain known as the hippocampus, and another one called novel object recognition, which you don't need a hippocampus to do this second task. This down here, this is two stylized images of the hippocampus. It's even when Golgi looked and Cajal looked at it, it was a beautiful structure just with Golgi staining. This is a artistic view of it, which was done by a former neuroscience student here at Penn named Greg Dunn, D-U-N-N. So if you would like to buy one of these images, this is what he does for a living. So gregdunn.com or something like that. But I warn you, they're not cheap. So, but they're very beautiful. So the hippocampus is, not surprisingly, a brain region that's critically involved in learning and memory, particularly spatial memory. So if you are pathologically losing your keys, you might have a hippocampus problem, okay? And then more seriously, people like Alzheimer's patients, pathology in the hippocampus is one of the first places it shows up. Okay, so we have two tasks: object place recognition, which depends on the hippocampus, and novel object, which doesn't. Okay, so what's this task? So the first thing we do, we just have an open field. We put the rat in there, allow him to run around for a little bit, habituate to the chamber. The next day, we put him back in the same spot, but now we have two objects which are absolutely identical. And we allow the animal to explore these two objects. They spend about the equal amount of time exploring each of the objects. Then the next day, we move one of them. We take this one, we move it over there, and the animals tend to explore the one that's moved more. Okay, so we've moved something, the spatial orientation of these things has changed. We said that the hippocampus was involved in spatial memory, so not surprisingly, this is hippocampal dependent. Novel object is similar, except for training, we've got the same two objects, but then during testing, rather than moving one, we just swap it out for a novel object, okay? And they tend to prefer the one that's novel, right? And you don't need a hippocampus for this. Okay, so then we ran our offspring of the cocaine dads and the saline dads through these tasks. And first, let's talk about novel object recognition. So if we look at the white bars, this is training. So this is during the period where they just get to explore these two objects. And I told you that they spend about the same amount of time exploring the two objects, and that's what we see, right at 50% through all the groups. Then the next day, we swapped out one of the objects, and I told you that they tend to prefer the novel object. So in this case, in every group, they preferred the novel object, okay? So the blue is for boys, and the pink is for girls. So this is, in every case, this is normal behavior, okay? So none of these animals, their novel object recognition is just fine. That's in contrast to spatial object. So again, females, so unlike Teresa's talk, in our case, the females are just fine. They do super. But the males is a different story. So in terms of the spatial object recognition, it is completely gone. So they have no preference of one for the other. They have no recognition that that object has been moved. They spend equal time. So we have a severe deficit in spatial object recognition. Okay. So the neuroscientists in the audience, if you saw this data, well, the first thing you'd say, well, everybody in the audience, hopefully you're thinking hippocampal deficit, right? Because I told you that this task was a hippocampally mediated task. All the neuroscientists in the audience then undoubtedly immediately thought, let's look at LTP, which is what we did. But first, I should tell everybody else, what is the LTP? So LTP is something called long-term potentiation. Long-term potentiation happens to have been demonstrated the year I was born, 1966, 47 years ago. So LTP is an enhancement in neuronal activity produced by electrical stimulation. So we put a little electrode down in the brain, we stimulate these electrodes, and the electrodes cause a change in the activity that's persistent. So changes in the brain, that's what we get with learning and memory. Learning and memory, by definition, results from a change in the brain. And LTP is widely considered to be one of the mechanisms underlying learning and memory, and not surprisingly, one of the easiest places to get it in the brain. And the first place it was demonstrated was in the hippocampus. Okay. So here we have in white, we have the saline-sired guys. In blue, we have the cocaine-sired guys. So over here is baseline. So during baseline, the cells are just firing along at their normal rate. And then here at theta, this is something called theta burst stimulation, but this is where the electrical stimulation came. So in the control group, then we get the potentiation. So this is the LTP response. Okay, so we have an enhanced neuronal response, and it stays up there. So we can think about normally during learning and memory, you're trying to learn something, and then during this, this process, you're consolidating those memories, and the hippocampus is deciding what it's going to remember, what it's not, and then it's going to store those memories. So the critical thing here is it is completely absent. If your dad took cocaine, you are incapable of spatial object recognition or hippocampal LTP. So this tells us, first off, it confirms that yes, the hippocampus is screwed up, and this is a physiological correlate, we call it, of the behavioral response that we observed. Okay, but I told you that changes in this case, the changes in the offspring are not that surprising. So in this case, you can imagine the dad took cocaine, but his sperm is also seeing the cocaine, and those sperm became these animals that we're testing here. Not so in the next generation. If you look at the grandchildren, they have never seen cocaine at all, in any way. The sperm that created them did not produce cocaine. So the next question is, what's happening with the grandchildren? So these are the parents. These are the children we just talked about. The girls are just fine. In both cases, if your dad took saline, you're just fine. And this guy here is the one that has the deficit. Okay, so now we're going to look at the grandchildren. And the critical groups, we just showed you these two data, and I'm going to show them again, data from these two groups, the cocaine-sired and the saline-sired male. And then in the next generation, the grandchildren, we're going to, if your dad took cocaine or your dad received saline. And basically, we show that the phenotype, and phenotype just means that the characteristic that we're observing is both in the children, so severe spatial object recognition deficits that are also there in the grandchildren, okay? So then, naturally, the next question is, what's happening with LTP? And this is a bad way to wrap up a talk, but we don't know yet. So our prediction is that they would have LTP deficits, but these are unpublished data. It's hot off the presses, so we don't know the answer to that question. But the bottom line here is that if you take cocaine, if you're a male and you take cocaine, you can be influencing the behavior and physiology of your children, and not only that, you're going to influence the behavior and physiology of your grandchildren. And who knows, it could go beyond that. My prediction that it will. But we don't know the answer to that last question. So as I said, Leo's in the lab. He was involved in this project. The lead author on this project will be Matt Wimmer. There he is. And everybody in the lab's got kids these days. Thank goodness I'm not having any more. These experiments scare the crap out of me. So everybody in the lab contributed, and I thank them, and I thank you for your attention.
Do you think that, like, as you said, that you think it's going to affect further generations, but do you think that the effects on the LTP will be as pronounced continuing the generations? Do you think it's going to kind of like maybe normalize or meet someone in the middle?
Well, so so my prediction with the the grandchildren, I expect the LTP to be gone because the behavior's gone. So so my expectation is that we will see the LTP gone in the grandchildren. And then the grandchildren, that indicates that there's a change in the germline, to get more specific about it. And if there's a germline dependent change, my expectation is it's going to be there with the great-grandchildren too. But you never know. These things could fade away, but the grandchildren data indicate that we do have a change in the germline, so it's heritable. So if it's heritable, I think it's still going to be there the next time.
Do you envision any future studies, or have read any that are maybe doing this already, regarding the actual dose-dependent effect? Like, how much cocaine does dad need to do to actually cause the effect, and what's the lower threshold to see these effects?
Yeah, it's a really good question. So in our lab, we study addiction, and most of the time in the lab, we study, as Leo said, animal models of craving and things. So we really think that the self-administration model is very close to what humans do, because as I said, we don't have to convince these guys to take cocaine. So that's a long-winded answer of saying I think we did really overkill in this case. I don't think that the animals had to self-administer the cocaine. I think if we put the cocaine in a syringe, administered it to the animals, we would have seen the same thing, because the critical thing in the dad generation is just that the sperm saw the cocaine. I don't think the brain's involved at all. I don't know that for a fact, but that's my guess. And so, so I think that we made it too complicated, but the amount of time is a good question. And I don't know. I mean, so 60 days is the length of spermatogenesis, which is why we chose that. We were very conservative. If sometime in the future we find out that 7 experimenter-delivered administrations of cocaine are enough, all my students are not going to be happy with me because it makes it a lot more difficult.
How specific do you think your effects are for cocaine? You know, would this generalize to other drugs or other addictive substances such as sugar or chocolate or coffee?
Yeah, I'm not touching those last ones. But there's clear evidence that this is new stuff. What we've been doing primarily is looking at the reinforcing effects of these drugs. So we do, the dads take cocaine or nicotine, or other labs have looked at things like heroin, and then they've looked in the next generations and seen the influences on drug-taking behavior. And it's too complicated to get into, but there are definite effects. They're not all the same. So not all these drugs of abuse don't all produce the same effects, but they're all producing transgenerational effects, including nicotine. And I've got bad news: if we have drinks afterward, it's alcohol too.
We'll have to take other questions for afterwards. Thanks again to that.